and this gives us the excretory unit. First, we looked at the many anomalies that may occur in the urinary system that may be as a result of a failure of development of the ureteric bud or an incomplete development of the ureteric bud. There are also other anomalies, of course, that may not be necessarily associated with just the failure of formation, but it may be as a result of mutations in some of these genes responsible for urogenital formation. I requested you to read about the woman's tumor and its associated anomalies, normally associated with certain anomalies. Today, we want to look at the genital system. Okay, I think I can now start on the genital system because we are about 40. So, the genital system also is derived from the intermediate mesoderm. The thickening on the posterior wall of the developing abdominal cavity that forms the urogenital ridge. Say the urogenital ridge gives us a nephrogenic ridge or cord, and that is what gives us the urinary system. And the genital ridge, medial to the nephrogenic ridge, that is what gives us the genital system. And when we are covering the genital system for better appreciation, we shall divide it into 3 structures. We shall look at it in form of the gonads, which can be the ovaries and the testes, then the internal structures, and these internal structures are mainly derived from either mesonephric duct or the paramesonephric duct. We may not so much use these terms. We may use Wolffian duct for the case of the mesonephric duct, and then paramesonephric duct. Instead of paramesonephric, sometimes we may end up using Müllerian duct. And then, of course, the external structures. The external structures mainly going to come from a condensation of the mesoderm around the end of the forming gut, that is the cloacal membrane. We partly saw the cloacal membrane forming a sinus that contributed to part of the urinary system. And today we are going to see a further condensation around the gut region, the cloacal membrane. But before we get into this embryology proper, we want to remind ourselves about sexual dimorphism. You remember from last semester too, when we were looking at cytogenetics, we looked at chromosomes, what a chromosome is, and we divide these chromosomes into the autosomals and the sex chromosomes. And generally, we recall that the Y chromosome determines sex, whereby if an individual has XX, then it is a female, and then XY, the male. So you remember things like the 46, X, Y, or 46, XX, or sometimes maybe 46, and then they add on this depending on how they are writing it. So when it comes to short dimorphism, we say that the Y chromosome determines sex, and what you see here is a Y chromosome. The Y chromosome is generally dominated by a male-specific region. Male-specific regions because this entire area has genes. Because if you divide this chromosome into segments, you could say that all these segments are carrying genes. So there are different genes along this stretch, but these genes encode for male-specific factors. Then there is a small region here, and then even on the cranial part of the upper arm, it is a pseudo-autosomal region, and we call it pseudo-autosomal region because the genes it carries can as well be present on an X chromosome. So in a way, it appears as if it is a universal chromosome, that this is not specific for the male. And what it encodes for is not really associated specifically to male characteristics or male development. Our bigger interest in the male-specific region is on a small region here, what we call the SRY region. So SRY is a sex-determining region on Y. What they mean is sex-determining region on the Y chromosome. And SRY is a gene that specifically encodes for a testis determining factor. So this testis determining factor is what governs or is what leads to the development of a gonad into a testis, and it further regulates the differentiation into phenotypic differentiation into the sexes. So where there is an RY gene, then you know that a testis determining factor is going to be formed. And where it is present, that gonad will automatically develop into a testis. And the individual will start later to develop male specific characteristics. So by default, if an embryo does not carry SRY, it means TDF cannot be produced. And by default, that individual will turn out to be a female, just because of a lack of this transcriptional factor. I hope you appreciate your transcriptional factor from your biochemistry. I think you've already done transcription and then translation. We should not get so much into that, but I want you to just appreciate transcription, that transcription factors are normally what regulate gene activity. That you're converting the message from this to RNA. So by the time the message is transferred from this to this, it has already been determined what this will form, what kind of proteins will be produced by this RNA, just because this transcriptional factor TDF has imposed itself on specific genes. So sex differentiation. Having looked at the Y chromosome, then we can ask ourselves, at what point during development is sex determined? When is an individual's sex determined? This is a question I'm posing to you. I'm glad Patricia has come. It was already here very early in the morning, and I'm not seeing some two people. Kataike? But I was expecting that actually the ladies are going to help us a lot when it comes to this area, given the fact that they always seem to have a vast experience when it comes to embryology of the genital system. So, Patricia, I would ask, at what time in life is sex determined? Okay, someone is saying my network is poor. Is it for everyone?
No, it's good.
Yeah, so I think it's an individual problem, not this is not a problem for everyone. Patricia, at what time is sex determined or an individual's sex determined? Ezelit, we are not hearing Patricia. Ezelit, at what time is sex determined?
I think at three
Yes, please.
at 3 months.
That's 3 months. Okay. Yes, please. Felix.
Sex determined on the point of fertilization by which type of sperm is fertilized in the ovum.
Okay. Yeah. So I actually think Felix is right on that. That at the time when fertilization is taking place, sex will be determined because fertilization is involving either an X and Y gamete to fuse or an X and X gamete. Remember the normal Mendelian inheritance. So that is when we are going to get male or female. This happens at the time of fertilization. But at the same time, We can also take as a his argument, though maybe the timing might not be, that might be really too far. 3 months is a lot of time, but we can also determine sex after fertilization. And that is why we normally talk about genetic determination of sex and then phenotypic determination. So genetically, sex will be determined at fertilization. But at this point in time, at fertilization, through up to day 1, day 2, week 1, week 2, actually week 6, you cannot differentiate this embryo typically. And that is why we normally talk about an indifferent stage. So at this point in time, the embryo is at an indifferent stage whereby you cannot phenotypically differentiate whether it is female or male, though genetically the sex has already been determined. And why you cannot do that? Because the testes, the ovaries have not formed anything. And it is these that are supposed actually to give us hormones to start developing into female structures or male structures. But at this time, the gonads have not formed. So it is important to differentiate those two. The phenotypic differentiation, which comes much later, but genetic differentiation occurs at fertilization. So from those weeks, we talk about in different stages because the embryos are indistinguishable. And at week 7, phenotypic differentiation starts. And at week 7, it's because at this week 7, we are going to see that our primordial germ cells, remember the PGCs, the primordial germ cells, we covered these when we are talking about spermatogenesis and oogenesis. So at this point in time, the primordial germ cells were not anywhere near the developing gonads. But at week 7, these primordial germ cells have migrated from their storage point and they are entering the gonads. So the moment they enter the gonads, the primordial germ cells that are coming will either be carrying the X component or they will be carrying the Y component. Please mute. Okay. So it is at week 7 that they are entering the formed gonads. Otherwise, before that, these gonads were all empty. The moment the primordial germ cells enter, then we start seeing other processes kickstarting, including hormonal secretion by these forming gonads. But it's also important to know that at week 7, even if phenotypic differentiation has started, you can hardly appreciate the sex phenotypically at week 7. So at week 12, external genitalia is forming. And so using external genitalia, you can be able to tell whether an embryo is male or female. And of course, at week 20, the external genitalia has fully formed. Note, and this is something I talked about earlier on, that in case of absence of an SRY, then by default, this embryo is female. Okay. So, how does the gonad form? We already said that the gonad, together with the kidney system, what we are referring to as the paired gland, form from the urogenital ridge. The genital ridge is contributing to the formation of the genital system, and this lies medial to the nephrogenic ridge or the nephric ridge. So what we are seeing here in, I don't know, this is brown. This is the genital ridge, and it's lying medial to the mesonephric ridge. It's important to note that even as these ridges are forming, this is the genital ridge. It's going to run along the length of the developing abdomen, mean embryo. And very important to note that it has an associated duct. Actually, the duct, on the contrary, the duct lies lateral to, yes, sorry, this is taking a bit of time, but I want to draw this because someone yesterday was asking about this. So, medial to this duct, there is another duct here in blue. And the same thing happens here. But then we also have, I've done this largely, divide it. So what we are looking at in blue is the mesonephric ridge. And the medial is the genital ridge. And these two have associated duct systems. So in blue is the mesonephric duct that we saw. It's the tube you can see here in blue. And sometimes instead of calling it mesonephric duct, it's going to be called a Wolffian. Take note of that. Then, still there is another tube. And now that is the paramesonephric duct. Para to mean besides the mesonephric duct. So besides the mesonephric duct, there is another duct, the paramesonephric duct. And sometimes we are not going to call it the paramesonephric, but we are going to call it the Mullerian duct. So there is a Mullerian duct system and a Wolffian duct system for those two systems. And these two play an important role in the genital system. Don't forget that the Wolffian duct is generally derived from the genital, from the mesonephric ridge or the nephrogenic cord. but it still has a contribution to formation of the genital system. So in this section, you can see at this point the mesonephric system that it's forming its so what you're seeing here is the mesonephric ridge or the nephrogenic cord, and of course the duct system that rotate. Then medial is the genital ridge, and this, of course this appears on both sides. So having seen those ridges, they are developing but they are empty. So there is already a ridge here and it is forming a gonad, but this is empty. Empty because there are no primordial germ cells. And where do we get these germ cells? The germ cells are derived from the epiblast, or what we call the primary ectoderm. Primary ectoderm because during the 3rd week of development, as the epiblast is forming the 3 germ layers, what remains of the epiblast is what forms the ectoderm, and that's why we call it the primary ectoderm. That it will differentiate into the endoderm, the mesoderm, and what remains of it is the ectoderm. So that's why they use primary ectoderm to refer to epiblast. So as the epiblast is differentiating into the 3 germ layers, there is a region or there are cells that are going to be given off from the epiblast, and these are primed to be primordial germ cells. They are going to leave the epiblast and they will traverse the entire length of the yolk sac. So what you're seeing here are primordial germ cells that have migrated from the epiblast and within the yolk sac. That is where the primordial germ cells will be stored up to around the 4th week, the end of the 4th week, when they start again to migrate back. So it's important to note where these cells are coming from, that they are coming from the epiblast. And remember, the epiblast is the embryo proper, but then temporarily they will be within the yolk sac. So around the 4th week, these primordial germ cells, they now leave the yolk sac. They leave the yolk sac and now they move to enter the forming gonads, but which gonads at this time are empty. So in our illustration here, we can see in yellow the primordial germ cells are migrating and they are entering gonads on either sides. And this will be towards the end of week 6. That the beginning of week 7, we already have our gonads and they have the primordial germ cells within them. So from the time of week 1 up to, yes, Marvin. Yes, Marvin.
Good morning, doctor.
Yes.
There's something that I was seeking clarity.
Can you increase on your volume?
Good morning, doctor.
Yes, good morning.
Yeah, there's something that I wanted to seek clarity on.
Yes.
what could be the exact reason as to why the primordial germ cells have to first enter into the, like, migrate into the yolk sac? Is it to give time for maturity or something like that? I don't know. What could be the essence?
Thank you, Marvin. Actually, the reason as to why they have to migrate to the yolk sac is not clear. It is not known. Though it is postulated that similar to what we see in the processes of oogenesis and then spermatogenesis, that at one point these cells tend to be frozen. You remember, this is very clear in oogenesis, where the cells are arrested in meiosis, and they are arrested twice, and that is kind of a preservation mode for these cells, similar to spermatogenesis, that cells are inactive up to the time of puberty. So, well, some people postulate that this is a mode of preservation of these cells, that they are kept within the yolk sac as a mode of preservation, but the actual function of them moving to the yolk sac is really not, research has really clearly brought it out well. So when you talk about the indifferent stages, in different stage, we shall talk about 3 indifferent structures. We shall talk about the different gonad, and later on, we see the fate of this indifferent gonad, and that will be the same case with the indifferent duct systems, or the indifferent internal structures, and the indifferent external structures. So in this indifferent gonad, the gonad cannot be phenotypically distinguished between male and female. All that we see within this gonad is proliferation of these strands, of the irregular strands, what we call the primitive sex cords. So the wall of these forming gonads tend to proliferate to invade the underlying mesenchyme or mesoderm. And in that way, they form these sex cords. They are actually strands. And sometimes they are referred to as medullary cords. And why medullary cords? It's because they tend to spring from the cortical area, the cortex, and invading the medulla. So sometimes they are referred to as medullary cords. And this is happening, or it is starting even before the primordial germs get in. So this will be looked at. It will be seen both in the male and the female, and that is why we call it an indifferent gonad. Okay, let's use a larger structure here. So you can see that the wall of this forming gonad is in black, and it gives off these strands to invade the cells within this gonad. This illustration is now showing us the primordial germ cells arriving from the yolk sac to enter the forming or the empty gonad. And the moment these primordial germ cells enter, then the process of differentiation starts. And we no longer call that gonad an indifferent gonad, but now it is either a male or female gonad. We either now talk of a developing testis or developing ovary. So what is the effect of the primordial germ cells entering the indifferent gonad? In case these primordial germ cells entering have the SRY gene, meaning that in case they carry the Y chromosome, then this gonad, indifferent gonad, is automatically going to develop into a testis. That the testis codes that we were seeing, or the medullary codes, they further develop. And then we see that the wall of this gonad tries to thicken and separate itself from the rest of the cords. So it starts to form a distinct tunic. If the primordial germ cells entering the gonad do not have the Y, meaning that there is an absence of SRY, so there is no way the testis determining factor is going to be formed. Then by default, it becomes the ovary. And what you are seeing as medullary cords, they actually start to regress. And instead, other cell clusters form from the wall of the forming gonad. And those cell clusters eventually form what we call the cortical cords. In another slide, we are going to see the actual effect of TDF and how TDF suppresses the processes that we see in the female. These are illustrations from Angman's Embryology. And we are looking at an indifferent gonad here. So primordial germ cells carrying SRY come in, and you can see the further development of the testis cords. So the medullary cords continue to develop, and you can see here the further development of those cords. And as they continue to proliferate, what happens is that they encircle, or they take in, the coming primordial germ cells. So you would get cords forming, then they are enclosing primordial germ cells. That at the end of it all, we are going to have these testis cords, the medullary cords, having a wall, and within this wall, there are primordial germ cells. These later on develop into what we call the testis cords, of course, and then we talk about this wall having the sustentator cells of Sertoli and the primordial germ cell. And of course, in between, what you're seeing in between these individual cords, we have another type of cell now, the interstitial cells of Leydig. And these forming cells are now that are going to be influenced by the secretion of the TDF factor to start having their own secretions, and it is those secretions that drive the formation of the testis. Very important to note that these forming testis cords now, they are not hollow. They are carrying primordial germ cells within them, but they are not hollow. They remain solid structures. They remain solid and not patent at all. So there is no connection between the testis cords and the duct system that was formed by the Wallensian duct and the tubules. So it remains solid up to puberty. And it's only at puberty that the testis cords start to form, to hollow, to form a cavity, and they will eventually give us the seminiferous tubules. And of course, seminiferous tubules will join the rete testis to join to the rest of the duct system. But before puberty, the testis cords remain solid. And this also shows you that before puberty, there's no way someone is going to produce sperms, that even if the process of spermatogenesis goes wrong, the sperms cannot move because there is no cavity here. When it comes to the female, it's kind of opposite. We saw, first of all, formation of medullary cords in an independent gonad. But with the arrival of the primordial germ cells, these medullary cords are no longer supported. So they regress and they break off. And another generation instead is proliferated. And these are the cortical cords we are seeing here. They form cell clusters being pinched off from the epithelium of the forming gonad, now the ovary. And as these cell clusters are pinched off, they also end up encircling or surrounding the coming primordial germ cells. So as primordial germ cells are coming, these clusters are going to form around them. And at the end of it all, you will have a cell cluster, and within it, there is a primordial germ cell. And of course, later on, these cell clusters form follicular cells. And this time we shall talk about our ovary having primordial germ cells surrounded by a layer of simple cuboidal cells. And this is what we shall eventually call the primordial follicle. And it is this that will kickstart the process of oogenesis. So still at this point, these formed follicular cells start to have secretions, which secretions are geared towards formation and further development of the ovary, and later on, development of the different internal structures into a female structures and external structures into external female genitalia. So let us look at each one of these individually. We said with the testis, there's going to be a formation of the testis cords. And the testis cords have primordial germ cells surrounded by sustentacular cells of Sertoli, and then the Leydig cells. In the ovary, we have cell clusters that have surrounded primordial cells. So we talk about the ovary having a primordial follicle that has the primordial germ cells and then follicular cells. And it is now a differentiated gonad. But why does these primordial germ cells do what is being done in the male? It's because of one important factor, the Müllerian inhibition factor. So when the SRY gene produces the testis determination protein, the testis determination factor is going to influence these two cells, the Leydig cells and the Sertoli cells that are formed, to have two secretions. One, the Leydig cells are going to secrete testosterone, and the testosterone is going to govern the development of the internal duct system or the internal structure development, and then development of the external genitalia. The Sertoli cells are also going to secrete a very important factor, the anti-Müllerian factor. And it's the anti-Müllerian factor that suppresses further development of the Müllerian duct system. So even when we were looking at two duct systems, the Wolffian and lateral to that, the Müllerian duct, presence of the anti-Müllerian factor or the Müllerian inhibition factor suppresses further development of the Müllerian duct. And that is why in the male, the Müllerian duct keeps on regressing. Whereas in the female, because there is lack or absence of the Müllerian inhibition factor, the Müllerian duct continues to further develop, and it will give us the internal genitalia. And this will take us now to the genital ducts. And we've already talked about presence of the two ducts, the Müllerian duct system and the Wolffian duct system. So these duct systems at an indifferent stage will appear in this manner. That we have the Müllerian duct to the Wolffian duct, and then we have an indifferent gonad. So this is the indifferent duct system that they both appear, and they are well developed at an indifferent stage. But because of the presence of the testis determining factor that is governing formation of the Müllerian inhibition factor, we see different fates. We already talked about the fate of the Wolffian duct. For better appreciation, we shall still include it here, since it has a contribution. So in the male, the parameronephric duct or the Müllerian duct system will generally regress thanks to the anti-Müllerian hormone. Sometimes vestigial remnants of this duct are present in the male, and that is the appendix, testis. So we shall see an appendix, testis here, which appears a persistence of the Mullerian duct in the male to leave a vestigial remnant. The Wolffian duct, and I will add on the duct and tubules, generally will regress at its cranial part. So the upper part usually regresses, but adjacent to the forming gonad persists, and Wolffian duct specifically will give rise to the epididymis, the ductus deferens, the seminal vesicles, and the ejaculatory duct. We may have vestigial remnants still of the Wolffian duct in male, and it would give rise to this paradigmis. It's just a vestige from this duct system. The tubules also would form a component. So the tubules form different ducts, and these are the different ductiles that we're seeing here that will connect the seminiferous tubule once they form from the testis course to connect it to the rest of the duct system. In the female, we see the opposite. Maybe I should use, let me use this better illustration. In the female, we have an indifferent stage here of the both duct systems. But remember, there is absence of the anti-Mullerian hormone. So our Mullerian duct will not regress, but it will further develop to give us the uterine tubes. And of course, its codomedial migration that it tends to move caudally and medially, and its fellow on the opposite side also does the same. It moves caudally and medially, and they meet in the midline. These normally fuse to form what we'll call a uterovaginal. Okay. Okay. So this fuses to give us a uterovaginal primordium or a uterovaginal bud. And it's from this that we are going to get uterus, the uterus here, the cervix, and part of the vagina, the upper part of the vagina. All this is coming from the Mullerian duct system. The lower part of the vagina actually has a different origin. The lower part is going to come from the dorsal aspect of the cloaca or the cloaca. So they have different origins, and the lower part is from actually 2/3 of the vagina. The Wolffian duct in the female, we know that it regresses, and it only leaves vestigial remnants, the porphyron and the paraphyron, and then it also leaves the appendix vestigialosa around the tip of the ovary, and then Gartner's duct, and his duct. So these remnants normally are going to persist along the length of this Wolffian duct. So wherever there was a Wolffian duct, there may be a remnant that persists. That's why we are saying that persistence of structures of this duct around this region are going to give us Gartner's. But then we also have, we may have a persistence of the tubules themselves that it will end up giving us the epopherons and the paraphorons. We are talking about persistence of the remnant to give us the appendix vesiculosa, similar to appendix testis that we saw in the male. And of course, these vestigial remnants many times are not going to be of any consequence, but sometimes these structures tend to proliferate and they become secretory, that they form cysts, that someone can get cysts along the path of this Wolffian duct. These cysts will not be confused with the ovarian cysts or with the fibroid cysts. That is different. This one actually sometimes called the para-ovarian cysts, where origins coming as a result of persistence of the duct system. So they can form cysts and they may be symptomatic. So how does the vagina come up? We saw the two duct systems, Malarian duct systems coming from either side to fuse to form a uterovaginal bud or primordium. This eventually cavitates to form the uterine cavity. So the uterus will form and it connects with the upper part of this Malarian duct, which form the uterine tube. And below here, it is of course continuing as the cervix and a third of the upper part of the vagina. But as it moves caudally, it still touches the dorsal ends of the cloaca, and the tips have factors that are going to trigger this part of the cloaca to form a primordium for a vagina. So it would form what we call the sino-vaginal bulbs. And these vaginal bulbs also cavitate, forms a cavity, and it will eventually give us the 2/3 of the lower portion of the vagina. So at the end of it all, we are going to have a vagina having an endodermal origin as a result of coming from a part of the cloaca which had an endodermal lining, and then a mesodermal lining because it was coming from the mesodermal ridge. So in a way, this is unique in the female, specifically in the female vagina, that they have a dual origin. What are some of these anomalies we can get as a result of formation of the internal genitalia in the female? Well, there are so many anomalies, and you are going to cover this in a much organized way in OBS-GYN. We will cover them as Mullerian duct malformations or uterine malformations, congenital malformations, and normally they are classified. If my network is poor, maybe someone should just speak out, because if it is a problem with one person, I think there we can continue, unless when it is a problem on my side. So you'll cover them in a much organized way where they'll be classified from class 1 up to class 4, 5, 6. Generally here, we want to describe some of those anomalies and why they may come about, what may cause these anomalies. But when you talk about these uterine anomalies or the Mullerian anomalies, it's generally because of a failure of fusion. My pencil has gone. So it may be a failure of fusion, that as these two tubes, they didn't fuse to form the primordium, or it may be as a result of fusion, they actually form the fusion. You know, once they come together, they fuse, and since they were two, since there were two, you would see a septum. So this would be the first tube and the second one, and in the middle there is a septum, and that is the septum that we saw regressing. And once the septum regresses, you then get a hollow, a cavity. So failure of fusion, failure of regressing of the septum, it may also be as a result of agenesis, failure of the Mullerian duct to form or to develop further. So if it incompletely develops, you may end up with hypoplasia, that you get an underdeveloped small uterus, and normally this is a combination with the cervix and or vagina, or it may be agenesis, that it has not formed at all. So in this classification, they actually now start with the malformations to do with agenesis, either hypoplasia or agenesis, and they would call that class 1. And last time we were looking at agenesis and the simple difference between agenesis and hypoplasia. So that agenesis may be vaginal agenesis. It may be atresia. So here they are showing us vaginal atresia. The uterus developed well, the cervix developed well, but when it comes to the vagina, there is atresia. It may be atresia of the cervix. The uterus is okay, the cervix is okay, but I mean the vagina, but there is atresia of the cervix. there are those that they may not even have shown us here. The uterus and the cervix form, and it ends here without forming a vagina. The complete agenesis of the vagina. It may actually be a complete agenesis of the cervix. The vagina, the uterus forms, and it literally gets into the vagina. And sometimes combination of these two, cervix and vagina, have not formed or have not fully formed. So that would be the first class. But there may also be what we call a uniconnuate uterus. So a uniconnuate uterus well, the illustration here only shows us bisconis, but in uniconnuate uterus, rather, it may be that there is formation of one horn of the uterus, or sometimes one horn of the uterus forms to connect to the cervix and vagina, and there is rudimentary a rudimentary horn has formed. So this rudimentary horn might either connect, or it might not connect at all, or it might connect and it's patterned, or it attempts to connect, but the horn itself is not patterned. And in the bisconis is actually talking about a double uterus. Double uterus, in the sense, maybe that is not the right term to use. I should stick to bisconis, because if I talk about double uterus is actually didiphys. But in the bisconis, it is actually the horn itself that you are forming two horns, and it's going to have two horns of the uterus that form, and then they eventually come to form, to open up into a vagina, either with a double cervix, or it can be a double horn. They are not showing any horn. It can be a double horn, and then as it comes in with a single cervix, so a horn here, and then there is another horn here. But it is very important to note that there is a fusion between these two. So there was an incomplete regression of the septum. In the double uterus, it is actually different. I don't like this illustration much because it doesn't really bring it out well. But when you talk about a double uterus, that is actually a class 3. You find a uterus forming, and sometimes with a cervix and a complete vagina. It has formed well. And then you find another uterus forming. Actually, there are two uteruses, this one, and they are fully formed with a separation of the myometrium, with the walls separate. Different from just a failure of regression of the septum. So that is the double uterus, different from the biconis. And as you're reading, you will realize that it's actually complex. It may be uterus, biconis, And this is unicollis. And the unicollis is here. It may be biconis, and it is bicollis, meaning that double horns, single cervix. Bicollis, it has double horns, but it also attempts to form double cervix. Double horn, double cervix. The most important thing I want you to appreciate here are the different forms that can form. You not really go into the classes, the classifications. The arcuate uterus, for example, it forms like any other uterus, maybe without any anomalies. The vagina is okay, the cervix is okay, but instead, the body of the uterus tends to form like a dimple. Instead of forming a smooth wall, it's not easy to draw, but it tends to form a dimple in here, inside the cavity, whether on one side or on both sides. The arcuate uterus or the arcutus. And there is another one that is also associated with a failure of a complete regression. All these are failure of complete regression of the septum, or a complete formation of an incomplete formation of the cavity. There is another one that we would call a T uterus or a T-shaped uterus, that the uterus attempts to form, but once it comes to cavation, it actually forms a narrow cavity, which is in this manner, that it's narrow and it appears to form a T shape, unlike the known cavity of the uterus, which tends to form a wider and kind of somewhat a V shape. This one forms a T shape, and this one is normally associated to the use of certain substances for treatment, maybe for abortion or for treatment of anything that it may eventually affect the development of the fetus, the T-shaped uterus. In your reading, you'll come across